Okay, so let's get started. So last class, I think we looked into the syllabus, right? So we'll just get started with our first chapter, which is the introduction to bioinformatics. What is bioinformatics, and what is the data which we'll be working with? And we'll start with a technique called microarray, right? So this is the PowerPoint I have, and I'll be uploading this on Moodle. And so you can have access to this PowerPoint from your Moodle. And I also have uploaded the class syllabus, right? So if you get chance, just look into your account and then try to download the syllabus and go through the topics which will be discussing in this class. So with bioinformatics, so bioinformatics, first thing which we need to understand is what is this data, right? So is it genomics data? Is it proteomics data? What is genomics? What is proteomics? So genomics, the definition is it is the study of organisms, genome, and uses the genes. It deals with the systematic use of genome information, and it helps to provide answers in biology, medicine, and industry, right? So genomics is genome, right, or genes. So genomics is study of genes. And the opposite of that would be proteomics, right? So proteomics is study of proteins. And it is a large scale study of proteins, right? So proteomics is just not studying, say, structure of one protein, but it is studying the structure of many proteins. It's a large scale study of proteins, right? And proteomics is much more complicated than genomics. More importantly, while the genome is the constant entity, proteomics differs from cell to cell and is constantly changing through its biochemical interactions with the environment, right? So genes, they remain same, right? So they don't change unless there is a mutation or any changes, um, which are very not much common. But then proteomics, there is a lot of changes, like downstream changes which are happening in these proteins. So it's difficult to study these proteins, right? So once you form your mRNA is translated to different proteins. So in this process of translation, there can be different things which can happen, right? So one protein can not be similar to the other protein, right? So so it's a changing, there are changing dynamics for proteomics. So it's difficult to study any specific protein. But there are some techniques which help us to like model some proteins or make some very specific changes which can be replicated, right, from a cell, from one cell to another cell. So it's a challenging field compared to genomics, but nevertheless, like we can study the proteins, and this entire field is called proteomics, right? So I would be interested to understand what is the difference between genomics and proteomics, right? So then there have been significant development in the data banks, right? So data banks are where you store the genomics data and proteomics data. And genomics data would be just the different organisms like humans, mice, rat, C. elegans, Drosophila, and you would have different genes for these organisms. And proteomics would be just to have, say, protein structures for these different organisms, right? So the studied genes and studied structures, there has been a significant growth, right? So as you can see in these two plots, the first plot here is for the number of nucleotides, right? So how many bases have increased. And the second plot here is for number of structures or protein structures. And the x-axis is the year, right? So it's 1996 to 2004. And in 1996, you had, say, around, like, I don't know, probably say this number may be 5 to 10,000, probably less than 5,000 nucleotides, which were sequenced and known for the DNA in 1996. And that number has increased to like 80,000 in 2004, right? So there is a very significant improvement in the number of DNA sequences which were sequenced and which were deposited in these data banks. And the same is true for the proteins, right? So in 1989, you had like, say, like less than 1,000 proteins structures which were known, and then that improved to like 30,000 protein structures in 2004. So the point is, it's a growing database. Every month or every possibly like every other month, there is a new DNA sequenced or new protein structure is being identified, and then this information is deposited into these databases or data banks. So in our next few classes, we will be looking at the data banks which do have this information, and then if it's a DNA sequence, what kind of information does it have? What is the format of that DNA sequence? If you have a protein structure, then what is the format of that protein structure, right? So we will look into these databases, but this is the data which is essentially used into analyses in the field of bioinformatics, right? So in bioinformatics, we mostly focus on genomics data, which is DNA sequences, and also mostly protein structures or protein sequences called as proteomics. Any questions so far?
So both graphs show so both are for the genomic and proteomic, even though the genomic is the genomic the one that's supposed to stay constant?
No, I guess both has to, like, so first graph here is for genomics, like genes, nucleotides, and both the graph needs to increase linearly, right? So, and then this second one is for proteins. So structures is for proteins, proteomics, and nucleotides is for genes or genomics. Does that answer?
Okay, so the first graph is genomics and the second one is the proteomics.
Yeah, exactly.
Okay, thank you.
So that is the development of genomics and proteomics databases, and we'll be looking into some of these data sets, and I will also introduce you to these databases, right? So what exactly is this database, and how to deposit your data, how to retrieve your data, and so on. So this is just a map marker, right? So this the black structure here is, as you know, it's a chromosome, right? So it's a chromosome, and on this chromosome, they have marked some numbers here, right? So these numbers, these are locations, and these are the locations for the base pairs where you know that there is a gene, right? So genes are packed into these chromosomes, and there are chromosomes with pairs, right? So if it's an Y chromosome, that is for males. If it's an X chromosome, it's for females. And on these chromosomes, you have these numbers here: red, black, red, black, red, black, red, black. So these are chromosome locations. So these locations correspond to some base pairs, right? Some bases. And bases, as we know, there are 4 bases. adenine, guanine, thymine, cytosine, right? And these bases form genes, right? So there are known 3.3 billion base pairs of human genome. Okay, so from this slide, you just need to know approximately how many base pairs are there in human genome. And the number is 3.3 billion bases, right? So 3.3 billion base pairs, right? So that is a lot of information. Let's on the scale of big data, right? So you have adenine, which pairs with thymine, and guanine, which pairs with cytosine, right? So ATGC. So, and there are 3.3 billion of these pairs, right? So 3.3 billion pairs. So adenine with thymine, guanine with cytosine. There is a lot of information there, and some of the information is they call it as some of these bases. They are not important. Uh, they call it as garbage genes or garbage bases, right? So because they don't have any function, but some of them do have function, and we know that there are approximately 20,000 genes which have the functions in humans. So the idea is there are a lot many bases here, there is a lot many genes here, and so people have tried to mark them on this chromosome, right? So what is the gene location? What is its chromosomal location? What is its function? What are its bases? But still, there is so much of information which we don't know. And so bioinformatics is the subject which tries to understand the novel genes, novel proteins, what are their functions. We can predict the function, we can predict the structures, so there are a lot many things which you can achieve using bioinformatics or computational biology. So the next slide here is just an example of a metabolic activity or a pathway, right? So you must have come across some of these figures where it depicts the cell structure, right? So you have the cell, and then you'll have the plasma membrane, and then you would have like a cytoplasm and then nucleus. So in the cell itself, there are many things which are um, which are regulated, right? So most of these things are regulated by, say, proteins or by mRNAs or by DNAs. And this is one example here. So this is just one pathway, which is called as folate dependent pathway, right? So there are n number of these pathways which occur inside the cell, right? So this is a folate dependent pathway, and as you can see, this is just one pathway. You can have like 100 different pathways which are regulated inside the cell. And in just one pathway, you can see that there are so many proteins and metabolites which play some role in the regulation of this folate pathway, right? So there are like these methionines, there are homocysteines, there are adenylyl homocysteines, there are tetrahydrofolates. So these are all the metabolites, and then all of these metabolites, they have to work in certain mechanism, right? Certain way, certain order. And then some of these, they require like some additional metabolites for their activation or for their deactivation. Some require adenosine triphosphates. Some require some proteins for their activation and so on, right? So this is a metabolic pathway for uh, folate metabolism, and this is also one of the fields or subfields in bioinformatics, right? So bioinformatics can be used to study the mechanism of these pathways, which genes cause which metabolites to say up-regulated or say down-regulated, and then what would be the downstream effect of these metabolites. So bioinformatics obviously it helps to understand genes. It helps to understand proteins, and it also helps to understand the metabolite or metabolic activity in these pathways. So obviously you don't have to remember this. This is just an example of this pathway, and I just wanted to let you understand the complexity, right, of this pathway, and then what things happen in some metabolism, right? So how can we measure the metabolic process? So in the previous slide, we saw that metabolic activity of folate pathway, right? Folate metabolism. So, so bioinformatics, it helps to understand these processes, and the way to understand is genomics to proteomics, right? So first you understand genes. Based on genes, you get to know which proteins may be affected. Based on the proteins, you understand which pathways will be affected, right? So you move from genomics to proteomics, right? So metabolic pathway consists of metabolites, which are uh, increased expression, decreased expression. So to understand those increase or decrease expression of those metabolites, you study genes, because everything is coming out of genes. So genes, they form proteins. Proteins influence downstream pathways, right? It can be metabolic pathway, metabolites. So you start with genes, then you study proteins coming out of those genes, and then from proteins you can study the pathways which are affected by those proteins. So obviously in bioinformatics, we have to work with some data, and data can be, say, humans, right? So humans or human cell lines, that would be your data sets. But there are also some other organisms which you can use for um, for for performing your experiments. One of the organism is called C. elegans, right? So this is just a worm. It's a very interesting organism because there is a very significant overlap between the genes in this worm and between the genes in humans, right? So this is one worm where there is a similarity between the expression of genes or the genes itself in this worm and in humans. So it's a very interesting and important model organism. Uh, if you see that there are certain genes which are affected in this worm, then there is a very good chance that they may be affected in humans also, right? So these are called model organisms. So one of them is C. elegans, which is this worm. Uh, Um, the other can be, say, Drosophila, right? Drosophila is a fruit fly. Um, or the other can be, say, mice, a rat, right? So all of these organisms, they have a very high similarity of their genes with humans. And so if you want to understand changes in humans, then you can conduct experiments on these model organisms and test your experiments, and then it's very likely that the changes happening in these organisms can be seen in also humans, right? So this is a an example of model organism. Say three model organisms which we use in the lab setting is or are mice, C. elegans, Drosophila, right?
I think I might have missed this, but what did you say? Like, in what ways, is it like similar to the biology of humans? Can you repeat that?
Yeah, so the genes in these organisms, they are very similar to humans, right? So if say humans have 20,000 genes, these organisms may have like similar 10,000 genes to humans. So if you want to study changes in humans, and obviously you cannot use humans because you cannot do your experiments on human beings. So so what people do is they test their experiments or any conditions on these model organisms like worm or Drosophila. And if your experiment says that, okay, these n number of genes are affected, but since there's an overlap between these 10,000 genes in these organisms and humans, it is very likely that the n number of genes which are affected in these worms would also be affected in humans. So these are called model organisms. So they are model for humans, and they can use, you don't need any compliances or any permissions to use these worms. So that is where you use them to understand changes in humans.
Okay, thank you.
So until recently, we lacked the tools to measure the gene activity. In 1989, there was an introduction of a technique called microarray by Stephen Fodder. And in 1992, this technique became commercially available, right? Okay, so now we are getting into a technology, first technique of our bioinformatics subject, which is called microarray, right? So I would be interested to um, for you to understand when was this technique introduced, right? So the year was 1989, and who introduced this technique? The person's name is Stephen Fodder, right? So Stephen Fodder, 1989. It's a very important technique because it is where the bioinformatics um, uh, got got its data, right? So this was one of the data sets which was used for analyses. Um, so and it it is a very effective technology to study the gene expression changes. But at this point, it is getting little, probably a little obsolete because there are newer technologies which have been developed. As we see that um, this technique was introduced in 1989, which is like 30 years back, right? Almost 30, more than 30 years back. So it's getting a little obsolete, but nonetheless, we do use this technique. It is cost effective, right, compared to other newer techniques, and it gives you a very good understanding of the gene expression changes. And it was developed in 1989, but then it was commercially made available in 1992, right? And at that point, it was new technology, so it was a little costly. Obviously, for like 30 years back, uh, things were not cheap. So, but it was made commercial in 1992, and people can use this technology for studying the gene expression changes. But then uh, most of it was started like say 15 years back, right? So 15 years back, people this started using it very often, and then on different data sets, different labs across the world, right? Different countries, because they saw the importance of this technique, and it at that point it was affordable. So I will show you a data bank, right? We'll go through a database, and you can see that there would be like a million records of uh, these data sets coming out of microarray technique, right? So so that's the introduction of this technology. It's called microarray, right? So if you just break this word micro array, right? So micro is small and array is a chip. So you're using a chip to study the gene expression levels, right? So micro is a small chip, array is a chip. So using a small chip technique to study the gene expression changes. And you need to know what year it was discovered or developed, who developed it, and then what year it was commercialized, right? So that's your microarray. It's just an introduction of microarray. So this is a okay, let's just move this figure here. Okay, so this is the picture uh, which is showing you this instrument, right? So this is the on the right side, you see this instrument here. It's a robotic chip instrument, right? So I'll explain you how this technology work in the next slide, but then this is just and a chip setting instrument, and it has a robotic arm here, right? So what it does is you have to you have to drop uh, you have to drop your cDNA on a chip, right? So you take your cDNA and then you drop it, just one drop on a chip. So obviously, when you are trying to drop it on a chip, every time you do it, there's a bias, right? There's a human bias. You can have like a less drop, you can have more drop, you can have uneven drop, right? So to avoid those biases, people have come up with this platform where you have a robotic arm, right? So robotic arm will take like say 1 micromolar of your cDNA, and it will drop 1 micromolar of cDNA on the chip, right? No less or no more. So it's quite precise and sensitive. So this is just one instrument which has a robotic arm and it has a chip somewhere here. So this robotic arm just drops like say a measured quantity of cDNA on the chip, right? So this is just to give you an idea of this instrument. And the picture on the left side here is this is called as fluorescence intensities, right? So once you drop your cDNA on this chip, you would see some fluorescence intensities. So obviously I'll explain you what it is, but then you just remember that this is a chip, right? The square box is a chip, and then the fluorescence is the yellow color, right? So more the yellow color, more is the fluorescence. Less the yellow color, less is the fluorescence. So based upon this color, you define or you determine the amount of expression. More the fluorescence, more is the expression of genes. Less the fluorescence, less is the expression of the genes, right? So each of these circles is your genes, right? So this is gene 1, gene 2, gene 3, gene 4, gene 5, gene 6, gene 7, gene, and gene N, right? So it is divided into columns and rows, columns and rows, columns and rows, right? So because it's a chip. So based upon the fluorescence, you quantify this fluorescence, and then more the fluorescence, more is the expression of the gene, less the fluorescence, less is the expression of the gene, right? So this slide just gives you some understanding of what is this microarray instrument. We don't have that at Claflin, right? We don't have this instrument, I believe. So we just try to collaborate with some other core facilities outside Claflin to get access to this micro. It's a costly instrument, and we don't use it often. So there are core labs like Emory, like USC, right? So there are other core facilities which offer this service, and which is what we use to do our microarray analysis.
Are all the fluorescents different genes? Like, do they each
Yes.
code for different genes?
Yes, exactly. So each of these fluorescence counts for different genes. So this can be like gene 1, this can be gene 2, gene 4, gene 5, gene 6. Each of these is different genes.
So how do they isolate each different gene? Because you said they use cDNAs. Is it that they'll take a tissue and make cDNAs from all the mRNA in the tissue and then somehow isolate all the individual cDNA so they can put them on the chip?
No, yeah. So the first part is correct. So what they do is they say there is a patient, and this patient has a cancer, a tumor. So what they do is they do a biopsy and they isolate the tumor sample, And from this tumor sample, they isolate the tissue, right? So you get your tissue from your tumor sample, and the tissue, you break down the tissue, and then you break down the cells. And in that cell, you would have 20,000 genes, right? So you would have 20,000 genes. So you convert that, you reverse that, right? So you convert all those 20,000 genes to all the 20,000 cDNAs, right? So you convert all the 20,000 genes to all 20,000 cDNAs, and that is something which you take and put it on this chip, right? So you would have 20,000 cDNAs which are on this chip, and they bind to the probes on this chip. So in the next slide, I'll explain you what are the probes and what binding happens. But this chip would have all the 20,000 or known cDNA cDNAs from that sample. Does it make sense?
So it's like the chip is already programmed with each cDNA that could be there from the known genes.
Yeah. So this chip has probes, and these are known genes. So these probes
I
are
see.
yeah, right. So probes, they are known genes, and when you put your unknown cDNA, it will go and bind to these known genes.
Okay, that makes sense.
Yeah. So next slide, I'll show you exactly like this is the technology, and then there are a few other slides which gives you more description on how it works. But at the end, you will get this intensity values, and based upon these intensity values, you know which genes are more expressed and which genes are less expressed, and which genes are not expressed at all. Awesome. So, all right. So I feel I want to go into the details of this technique, and once we do some discussion on this technology, then we will go into the data bank, and then we will look into what is this data, and then how can we download this microarray data, and then how can we analyze it, right? So the last thing which I want to do is today, do you guys have a GitHub account?
I already followed you on GitHub, so
Yeah, me too.
Oh, did I show you in the last class, right?
Yeah,
yeah.
you showed us like how to like set it up and follow you. I think we have to go over like the how to submit assignments though.
Okay. Yeah, I see your names here. Okay. All right. So, so probably in the next class, we will go through a, microarray data set, and then I'll show you how to go into your repositories, how to create a new repository, and then how to upload your files in that repository. Okay, so this is good. I have your names here. I have your attendance. All right, so I think this is it. That should be it for today. So we are meeting again on Wednesday, right?
Yeah, on Wednesday. And so everything we went over today, is this the first chapter of our textbook?
This is just the basics. So I just picked up the things which may be important to understand before we go into the microarray technique.
Okay.
Yeah. Let's just try to just follow this PowerPoint, and then I will I'll give you the chapter names later, right?
Okay.
And sir, I have a question. Is this are we gonna do phylogenetic analysis in this class, like maximum likelihood and parsimony and Bayesian analysis? Is that this class?
We may not do that, but we will start with microarray analysis in R, right? So we'll be using
Okay.
R. I'll show you. You would have to download the software for you, and then to analyze the microarray data, we would need the R package and some dependency libraries, but that is the analysis we will start with.
Okay, sir.
Yeah. Uh, right. Any more questions? Good. So let's do this. I will upload the.
All right, have a nice day.
Yes, please. Okay. So, right. So I will upload this PowerPoint on Moodle, and then on Wednesday, what we will do is we will finish up this PowerPoint. Right. There are some slides here. We will finish up those PowerPoint slides, and then we will go into the gene expression omnibus data bank, and we will try to get some microarray data, and then we will set up your R software, and then we will analyze that microarray data using R. Do you guys have a laptop computer?
Yes,
Yes, sir.
and I was gonna say, so I have like the application R Studio. Is that what it is? What we're using?
Do you have this R?
Oh, okay, that one. The one I have is called R Studio, but but you say we're gonna walk through how to set up R anyway.
Yeah.
Okay.
I'll walk you through the software. Yeah.
Okay. All right.
Well,
Yeah.
what is the operating system?
Sorry.
What is the operating system?
Mine's Windows 11.
Windows.
And I have a MacBook Air.
Okay, so I have MacBook. So Mac, you have a different R version, and then Windows, you have different R versions. So probably in the next class, I will give you the links for the recent version of R, Windows and Mac, and then we will download it, and then hopefully it should work. And make sure you use the same computer for this class throughout the semester, because we would have some dependencies to install. And if you change the computer and then if you reinstall it, then you would have to reinstall the dependency, which I don't want you to do. So make sure you just work with one computer and have that one software and all the dependencies in the same computer, right? And yeah, I think that should be it. Awesome. So I think we are done. If you don't have any questions, I'll see you on Wednesday, same time.
Okay, thank you.
All right.